Ever wonder how you're able to get out of bed in the morning? How you're able to walk outside your house, or how you can make that game-winning shot?
You might know that animals get their energy from the food that they consume. But how do the sugars they intake turn into energy?
It's all thanks to a process called cellular respiration.
Cellular respiration is the harvesting of energy from sugar molecules and the storage of potential energy which cells use to do work.
So what does all that mean? Well, basically, it means a sugar molecule, say glucose, enters a cell and is broken down through three processes. These processes convert the glucose molecule into ATP, or adenosine triphosphate, which serves as the main energy source for the cell.
Okay, but what are these processes and how do they convert glucose molecules into ATP?
Well, since you ask about these different processes, I shall tell you. The first process, glycolysis, breaks down glucose molecules and converts them into pyruvate molecules. This enzymatic process turns the 6-ring carbon molecule into 2 3-ringed pyruvate molecules. This is done in the cytoplasm through 9 different complex enzymatic steps. Not only does glycolysis create 2 pyruvate molecules, it also results in the production of 2 ATP molecules from 2 adenosine diphosphate molecules and 2 phosphorus molecules, as well as 1 NADH molecule from 1 NAD molecule per pyruvate. The NADH molecules hold electrons, which can be used later in the cellular respiration process. While 4 ATP molecules are made, glycolysis must use up 2 ATP molecules in order to run itself. Thus, the net product only yields 2 ATP molecules per glucose.
So far we've learned that one glucose molecule is broken down into 2 pyruvate molecules via glycolysis, which makes a net product of 1 ATP molecule. But our cells need way more ATP than that to function. So, how else do our cells create ATP?
I'm glad you asked. Well, the next step in cellular respiration occurs after 2 pyruvate molecules are formed. From the cytoplasm, we now enter the mitochondrion. Here, pyruvate is enzymatically converted into acetyl-coenzyme A, releasing CO2 and reducing another NAD to NADH. This is called pyruvate oxidation. This leads us to the citric acid cycle, which is also known as the Krebs cycle for its discoverer Hans Krebs. It basically uses acetyl-CoA to reduce 3 more NAD to NADH and to convert an FAD into an FADH. These electron carriers will be used in the next step of cellular respiration. The Krebs cycle also enzymatically converts ADP and a phosphate into ATP. It does this once per acetyl-CoA.
Okay, so now per glucose molecule, we have made 2 ATPs through glycolysis and 2 ATPs through the Krebs cycle. I'm sorry to tell you, but our busy cells are going to need more than 4 ATP molecules per glucose to function.
Hold your horses. We haven't gotten to the final step of cellular respiration. Presenting the electron transport chain. Remember all of those electron carriers we produced before? Here's where they're going to become useful. Throughout glycolysis in the Krebs cycle, 10 NADH molecules and 2 FADH molecules have been made per glucose molecule. Now, they will be able to provide the electrons that will be pumped down the electron transport chain. Once an electron is dropped off, a mobile electron carrier will move it through the inner mitochondrial membrane. As each electron passes by an integral protein, a hydrogen ion, or simply a proton, from the inside of the matrix goes through the protein and is pumped into the inner membrane space. And as this continues, too many protons will be in the inner membrane space and must get back into the matrix, thus going down their concentration gradient.
Now, just because protons are going down their concentration gradient, how does that make ATP?
Patience, silly. We're getting to the best part. The protons get back into the mitochondrial matrix through a protein called ATP synthase. As they do this, ADP and a phosphorus molecule combine and ATP is synthesized. Just like that. This process is called chemiosmosis, which is when the potential energy of a concentration gradient, in this case of the protons, creates ATP. Remember before how we made only 4 ATPs from glycolysis and the Krebs cycle combined? Well, the electron transport chain can be used to make up to 34 ATPs.
Yes!
Yeah, another common name for this process is oxidative phosphorylation. This has been a Wise and Son video production.
Check us out next time for an in-depth look at photosynthesis.
Bye! Ology!